Well, well, well. Well, hello there, everybody, and welcome to GCSE's Day 1. Yeah, bloody hell. So, the RE exam, let's talk about it. It was good, I think. There were only maybe one, two questions that kind of annoyed me. There was that question about how Muslims struggle to worship, struggle to practice in the modern world. That was a bit of a scummy question because I couldn't really think of any teachings. But it turns out a lot of people talked about like the pilgrimage to Mecca and how that was like expensive. I totally forgot that you could have talked about that, so maybe didn't get all the marks for that. Then there was the Eid al-Adha or something like that, the festival after Ramadan, and I completely forgot what it was, so I made up two things. So, in conclusion, a decent exam aside from that. On a scale of 1 being it was shit and I'm gonna get a 1, to 10 being like, this is great, I'd give it an 8, a 7.5. But anyways, we're not here to talk about RE. Let's do some biology revision. This is important, guys, because biology is tomorrow. I know a lot of people will click on these streams, comment something, and then click off, but seriously, guys, this is your last chance now to revise for biology. Because, I mean it now, it is happening very quickly, very fast, very soon. Go check the Google document if you don't have the time to watch this full hour stream. Please do. The Google Doc will contain everything you need to know on biology, chemistry, English, math even. But let's get right into this. So, topic number 1, you have the cells. It is. Kieran, if you have anything that you want me to go through or anything, anything really, let me know. Pick a topic really, because I'm just gonna start from basically the easiest thing I know and work my way up. So if you want to kind of jump the gun, let me know. We have the structure of a plant, and I'm gonna draw a palisade cell. Everyone always forgets the names of palisade cells, but they're basically the plant cells that you learn in year 7. Now, the palisade cells are right at the top of the leaf. If we have our leaf here, something like that, maybe there's another couple there, something like that. I don't know. Assume that they're on the top, basically. I forgot about palisade. Well, yeah, the palisade is the one that a lot of people forget, I would argue, because it's so generic. It's the most generic one. But you need to know the adaptations of this. So you want to talk about the fact that it is right at the top of a leaf. In the structure of the leaf, you have... I can't exactly recall the exact layers, but if I'm not mistaken, it's the epidermis, the skin, the waxy cuticle, so waxy cuticle, epidermis, palisade cells, spongy mesophyll, and then, right at the bottom, it is the guard cells and the stomata. That's how it works. So the palisade cells are right at the top. Why is that? Well, the palisade cell's function is photosynthesis. I guess you could associate the P in palisade with photosynthesis. It has a shit ton of chloroplasts, which means it's going to be absorbing the max amount of light. You could even in the exam talk about their wide surface area, which is needed. In an exam, try and incorporate surface area into your answers as much as possible, because surface area is very important to a lot of biology. And I'll include some examples as we go on, but that's the palisade cell. Right at the top, bunch of chloroplasts, a vacuole. I believe this is just for I'm not sure. Do any of you guys know what vacuole's purposes. I always just know it that it stores sap, but I'm sure there's another use for it, about osmosis or diffusion. If any of you guys know, that would be really interesting. I like to know. But anyways, the waxy cuticle is a physical barrier on the top of a plant to prevent water from staying on the top. So imagine like you know you've had a shower or something, and this is how people get like fungi in their feet, like a fungal infection. It's because there's water like in your toes that you haven't washed. Same kind of goes for plants. If there's water droplets on the top of a plant, what's going to happen? Well, fungal growth. It's going to get infected. The waxy layer prevents the water from penetrating, and it also just will make the water fall off. That's the purpose of a waxy cuticle. It's a physical defense mechanism. Okay, the next one, the epidermis, the skin. That's kind of all you need to know. Epidermis is skin. When we're looking at a microscope, we're looking at an onion epidermis in the practical. That is what we need. Speaking of practicals, there are a couple that I don't think we've gone through with Mr. Hooper that I will go through today, so you guys are aware. The first one is the onion epidermis experiment. So it's where you get an onion, you'll use a scalpel to cut into it. Hello, sorry about that, bit of lag. Anyways, like I was saying, so you take the onion, you'll use the scalpel to cut into it. You'll add it to a slide. Then you will add a stain, basically an iodine solution. But in my case, I'm using food dye for like my microscope, but the preferable term is a stain, which actually makes the cells, because cells are transparent, this makes the cells more visible. I'm not sure what the exact terminology would be there, but I guess you could say it allows you to see the cells. If you get a question on why you would use iodine, it allows you to see the cells, because cells are transparent, so you need to like dye them a colour. Then you would add your cover slip on, then you would place it under a microscope on the stage. Now, a thing a lot of people forget is the job of the knob, the adjustment knob. It literally, I have a microscope, I know this for a fact, it moves the stage up and down, but you can't say that. You have to say that it sharpens the image, I believe. I believe that is the terminology you would use. It sharpens the image, allows you to see the image clearer by sharpening it. You could actually drop marks in an exam by actually giving it proper definition. So that is the first practical. Many more will do. So, let's keep on going about the plant. You have the root hair cells. I just draw them like a big T, and then I kind of draw like a random mesh in the middle, and I have some mitochondria scattered around, because the root hair cell's function is to absorb water and mineral ions. And mineral ions enter the plant through the roots. Aha! Root hair cell. That's obviously where they are. And they go there through active transport. And active transport is not passive, it requires energy, which is why they have a shit ton of mitochondria. What's next? I guess the stomata and the guard cell. I'm not sure how you draw guard cells. I've never really seen them like drawn. I think they're kind of like elongated like this in a way. They probably have like a vacuole in, maybe. I don't know. They're a plant cell. Of course they're going to have a vacuole in. Maybe they don't. Who knows? But anyways, they basically open and close to regulate the opening and closure of the stomata, which are little pores within the leaf, which allows gases to enter. Although I guess technically water vapor. So for example, carbon dioxide would go in, because that is required for photosynthesis. And then waste products such as water vapor and oxygen would be released through the stomata. Very cool. Next is the xylem and the phloem. These things I used to never understand, but now I do. They are very, very funky things indeed. So the xylem and the phloem. So you might get a 6-mark thing in the exam discussing the differences between these. Consider them the blood vessels of the plant. But that's kind of how Hang on, let me go check something. I'll be right back. Mr. Beast! Alright, as I was saying, the xylem and the phloem. Okay. Let's state a couple of differences between them. You guys can put this in chat now, I won't look until I've studied them myself. A xylem is made up of dead cells. They are strengthened with lignin, and they are part of a transpiration stream. Because they take water from the roots to the leaves. Very nice. The phloem are made up of living cells. They have pores which allow sap to leave. They are part of translocation because they move glucose from the leaves, where it is created in photosynthesis, to elsewhere in the plants. I guess a final one, if you guys wanted to add even more detail, and Chewy, you hit every single one, you could talk about how the xylem is one way, the phloem both ways. And there you go. How many of those did you guys know? Let me know now. Because remember, you may not get any questions on the xylem and the phloem, but it's just, I think it's fun to remember anyway. It's just the blood vessels of a plant, you know what I mean? Fun. So, if you guys want me to move on to something else, let me know. Yeah, it's very nice. If you guys want me to move on to something that you want, let me know in the chat. But if not, I'll just move on to blood vessels and the heart, because I saw that TJ was stressing about this. I saw that a lot of people, maybe even Ashton, was stressing about this. I personally don't think that the I don't think that the heart is bad. Do you guys think it's bad? Who knows? Maybe I'm missing something here. I don't think it's too bad. I always draw the heart like a box because it helps me remember it. You obviously have it right and left. These two blood vessels going in, going out. TBH, yeah, like, I think the reason it stresses people is they have to remember the names, but likelihood is, in an exam, you'll get maybe 2 questions on the structure of the heart. You'll likely instead be asked a question on why, what does the heart do? What happens to it? You know, like, what, things like that. That paper that Mr. Hooper made us do today, that big one, was the best paper I've ever seen because it is so interesting. So there were questions there on the heart, how cancer can affect the blood. There were questions on the gallbladder, all stuff that I really am good at. So that was a godsend of a paper. If I even get something like that in the exam, I'll be crying. I'll be happy. That's why I learned the heart and the digestive system very well. But anyways, you have, going from right to left, you have the vena cava, the pulmonary artery, the aorta, and the pulmonary vein. You have the atriums and the ventricles. Very nice. But now, let's get into some actual exam questions. Where in the heart are the cells that act like a pacemaker found? Whereabouts will they be? Because remember, there are cells which, like, I guess, create a heartbeat, technically. They would, of course, be in the right atrium. Pacemaker cells. And pacemaker cells, of course, give you a heartbeat. The heart contracts, which allows blood to flow through it. But what if you have an irregular heart pattern? Well, that could lead to you needing an artificial pacemaker. Now, in terms of heart diseases, you could, of course, preventative measures for this. Well, first of all, let's talk about causes. If we assume that this is one of your arteries here, how is heart disease caused? Well, obviously, it's getting clogged up because you're a fat shit and you're eating fat food, innit? You're a fat shit. So yeah, coronary arteries. Too much lipids causes a fatty buildup in the coronary artery. Pair this with lack of exercise, and smoking even, and you're fucked. So your coronary artery gets built up with fat, which eventually stops the artery from being able, or it reduces the blood flow. What will this do? Well, if blood is completely stopped, you would have a heart attack because your heart would just completely stop. Or, alternatively, coronary artery, if it's slightly, you're going to feel a bit more tired because there's going to be less blood going around your body. Right? Less blood going around your body, which means less oxygen for respiration. You're going to have less energy. And maybe that's a leading sign of heart disease, fatigue. I don't know. You guys can let me know there. But that's my theory. But anyways, 2 ways to prevent heart disease. You could use stents or statins, which I'll just draw like a pill. So stents. Stents are, of course, like little coils of wire, which widen. So remember, the inner bit of the vein is called the lumen. So they widen the lumen, which allows for blood to flow instead of it being blocked up. Some advantages of stents is that they're usually a permanent or somewhat permanent procedure. You can't forget to have a stent. It's permanent, once you have it, it's stress-free. Some disadvantages of a stent is that you could have it rejected. Your body could reject it. It's an invasive procedure. You could get infected. It's a bit of metal after all. Those are some disadvantages. So statins, in case you guys don't know what cholesterol, I think that's how you pronounce it. It's basically like bad lipids. Right? That's what I found, basically. They're lipids which I think take more energy to be broken down, or like they're more dangerous to a person. So lipids are fats, okay? So this reduces the quantity of those bad lipids. In other words, they're quite reliable. So some advantages of statins is that they're non-invasive, your body won't reject it, and it is relatively effective at, removing lipids. The issue is that you have to take them for a long time, maybe the rest of your life. I know that I think both my grandparents, and I think my dad have to take them, which is a bit concerning for me. That means I most likely have heart issues when I'll most likely have heart issues when I'm older. Which is why I do Creed Rise to Glory boxing all the time. I want to make sure I have a solid, exercise. I don't want to be sat, fucking obesity and shit. I want to be good. Anyways, statins, the disadvantages, you could forget to take them. And they might be expensive. Although you shouldn't talk about price in the exam. That's the number one rule. If you mention price, I'll put a price on your life. Now, let's go to the blood vessels. You of course have the artery. Well, I've made the artery there very obviously an artery by making it have a super narrow lumen. I don't think that's how it is IRL, but it's a diagram in it. So, let's talk about the differences between these. The differences and the similarities. Number 1. These, in terms of lumen, have a narrow lumen. These have a wide lumen, veins. Arteries have a thicker layer, a thicker elastic and muscle tissue. in order to withstand higher pressures. Veins have a less tissue. Arteries are 1 cell thick. Veins have valves you could talk about to prevent backflow. You could talk about how arteries go out of the heart, veins go in. I feel like I'm missing something there. Am I missing a difference? Or I think that's it, really. Capillaries, of course, blood. They allow ions to diffuse in and out. Capillaries are really good. You could get a lot of questions on the capillary. They appear in the villi, the alveoli, that's what you call them, and gills. So there you go. That's a whole mind map kind of thing about the heart. If you didn't know any of that, know it. Let's quickly do adaptations. So this is meant to be kind of a villi type thing. Why am I drawing squiggly? Because these are meant to represent capillaries. Capillaries are 1 cell thick. They allow for a constant blood flow. Why is that? Well, a constant blood flow, constant fresh supply of blood means that there is always a strong diffusion gradient. Things can diffuse into the blood very easily, and then via active transport, they are able to leave. Or via diffusion. You could also talk about, in terms of the villi and the alveoli, large surface area. You could talk about that. The fact that the alveoli are 1 cell thick, which means that, air is able to go through. In terms of the alveoli, you could also talk about the fact that, the villi have a lot of mitochondria for active transport, so that there is constantly ions going through, even if the concentration gradient is a bit skewy. Anyways, the alveoli have a layer of moisture, which allows the oxygen to be, kind of dissolved as a gas. They also always just have a fresh supply of oxygen because the lungs are there. So their function is always being taken. Alright guys, what should I go on to next? I don't know if Kieran's still watching. I'm going to go grab a drink. So we've gone through somewhat the digestive system. Let's actually do one more bit of the digestive system. Actually, no. Let's loop it back quickly to another required practical. The pondweed. This one's easy. Just get your pondweed in a test tube. Put it in a glass beaker. You would get a ruler, like so. You would get a light at the end of it. Turn the light on. Wait maybe a minute, 2 minutes. Count how many bubbles are produced. Or, alternatively, you could get a gas pump, and this would measure for you the amount of oxygen produced. Maybe in an exam, if it asks you how to improve this reaction, you could talk about using a gas pump. So, you could talk about how this light should be an LED, otherwise it will release heat, which may, if it's not a limiting factor, if temperature is a limiting factor, more bubbles will be produced. But if it becomes too high, the enzymes will begin, you have to say begin, enzymes will begin to get denatured, which will make their active sites no longer bind, making less oxygen bubbles. That's for practical. In an exam, you may get asked how to make a practical better. You should always simply talk about repeating multiple times in order to calculate an average mean and plot it on a graph if needs be. So, let's loop it back to digestives. Let's talk about the gallbladder. Gallbladder is for bile. What are the functions of bile? Such a horrible word, I hate it. But bile emulsifies fat. Now, what does that mean? Well, let's assume we have like a fatty molecule here. It's going to get emulsified. It's going to get broken down into smaller bits. What will this do? Surface area. It's all about surface area. This makes the surface area larger overall, meaning that the enzymes are able to break them down into the useful products quicker. Bile also has an important part to play in stomach acid. Stomach acid. Your stomach makes acid, I think a pH of 2. This is bad. You don't want to drink that shit. But bile neutralizes this pH, which is useful because what if your stomach acid, hypothetically, got into like another part of your body? That would be shit, and also that would denature your enzymes, which would prevent you from being able to, have food. Bile is essential for life, kind of thing. Bile, there is such a good question about cancer, a bit morbid, but bear with me. Basically, the question was on cancer and why a person may have trouble digesting fatty foods if cancer is blocking their gallbladder. That question is piss easy now because we know that without bile, fatty food is going to be unable to be emulsified, meaning enzymes will take longer to break it down or will be unable to break it down. Additionally, bile will be unable to neutralize the stomach acid, which means that the stomach acid could cause enzymes to be denatured because they're not at an optimum pH. They're not at the perfect pH. Alright? Let's do food tests and enzymes quickly before we move on. We'll do infection and response. Then I think we're done, right? Are we done? Organization. Cell biology. I think we're done. You guys obviously know the I am equation. You guys should know that off the top of your heart. It's easy as shit. Anyways, let's go do our food tests. You got Benedict. Bayou Rare. You got Sedan. And you have iodine. Now, we could link this to ecology, not ecology, but plant cell and stuff, but give me a minute to do that. Okay. Benedict's solution is testing for sugar or glucose. I saw even Adam was confused by this question today. It talks about how you would test for glucose. I didn't even know this first time I got asked. Glucose is sugar. Just remember that. Bayou Rare is for protein. Sedan is for lipids. And iodine is for starch. Now, what are their colours before and after? Well, Benedict's will go from blue. But Benedict's will go from blue to red, orange, or green. Depends on the concentration of glucose. Bayou Rare will go from blue to purple. Just remember that as purple protein. Sedan free, it is originally red, but if there is fat, if there is lipids in, it will separate to the top. This red will separate. And finally, iodine is a brown colour, but it goes completely black or blue if starch is present. Just for that one, remember Kieran's bars, leaving you black and blue like starch solution. There you go. A quick note, Benedict's solution requires it to be in a water bath of over 65 degrees Celsius or more. And that's kind of it, I think. Now, how could we link this to the plants? Well, think about it. If we wanted to test a plant, we could possibly get a plant and cover a little bit of it with black tape. After maybe a week, we could take the whole plant, crush it up, or maybe, let's assume we cover the whole plant in black tape. After a week, we're going to go put some tests on. We're going to crush it up and use this kind of liquid, this pulpy liquid. What tests could we do? Well, we could do an iodine test or a Benedict's test. We could do Benedict's to work out glucose. If there is no glucose, that obviously suggests that it hasn't photosynthesized, meaning that there is no light, because obviously it's been covered up. Iodine, we could use to test for starch. If there is no glucose, there won't be any starch, because starch is glucose stored. Remember, the uses of glucose are respiration, fats or oils. The other uses of it are obviously starch, like we just said, and protein synthesis. So remember, iodine and Benedict's you could use. Just remember, you may need to add I think it's an acid. You mean ethanol. You will need to add ethanol if you're going to just get the leaf and add it to the test tube. Why? Well, we need to break down the cell walls. The cell walls will prevent something from bursting. Cell walls are thick, so they're going to prevent, the solution from getting in. So you need to break that cell wall down. Let's do enzymes, and then we'll wrap it up with maybe 3-4 things that I know a lot of you guys don't like. Enzymes, you have amylase, protease, carbohydrates, and finally you have lipase. Very funky names. Okay. Each one of these can be found in different parts of the body and turns into something different. Like magic. Well, no, they turn the substrate into something different. Amylase can be found in the salivary glands. So too can carbohydrates. All of these, I believe, can be found in the small intestine. Correct me if I'm wrong. Protease can be found in the stomach, and all the others can be found in the pancreas. I believe lipase is the only one that is found exclusively in the small intestine. But just to make sure I'm not spreading misinformation or anything like that, I will go ahead and check that myself. Because I feel like I may have slipped up there. Have a look. Aha, they are found in the pancreas too. So there you go. Now what amylase converts starch to glucose. Proteins to amino acids. Carbohydrates is obviously carbs, and that goes to simple sugars. And finally, lipase turns lipids into fatty acids or glycerol. So there you go. I'm back. If you don't remember me, I was dapper. Not to sound rude. I can't see profiles or anything, so I'm not too sure if I recall. But hello, welcome to the stream. We're just doing biology stuff because GCSEs are happening now. So let me quickly do hypertonic, isotonic, and hypotonic. Isotonic is when there is an equal concentration in and out, so in the cell and out of the cell. Hyper is when there is a higher concentration in the cell, which means it's going to shrivel up due to diffusion, osmosis, sorry. And alternatively, hypotonic is where the cell is going to end up like Pyrocynical's fan art. It's going to end up bursting because there is more water outside, which means more is going to go into the cell, causing it to burst open. Oh, I think I do know. That name does ring a bell. That name does ring a bell. So that's hypotonic, that's isotonic, and that's hyper. They're a tad annoying to remember, but once you kind of got it, you got it. Okay, next thing I'll go through is trying to think what stumps me in exams. I mean, we could do infection and response, but this is relatively calm. You got like your pathogen, you got your lymphocytes, you got your phagocytes. Phagocytes engulf them, lymphocytes attach antibodies to the pathogen, which may cause them to like connect together, which makes it easier for the phagocytes to engulf them. The way the phagocytosis occurs is it basically surrounds the pathogen, it releases enzymes which basically kill it. Lymphocytes also release antitoxins which bind to the toxins that like bacteria may release, which makes them, obviously the toxins are neutralized, they're not going to work. So the way that vaccines work is a dead or dormant version of the illness is going to be sent into the body, injected. This will trigger an immune response, which will cause your phagocytes to become active, your lymphocytes to become active. After defeating the pathogen, your lymphocytes will now have the correct antibody that they have used to just defeat this dead pathogen. They will use this antibody in memory lymphocytes. They will store the correct antibody. Next time you're infected, that memory antibodies are going to produce en masse, which produces the antibody en masse, which kills it very fast. Okay, finally, I say finally, I probably missed a lot, so let me guys know if you want me to go through anything. Again, I'm not perfect, I'm not Jesus, I've probably made a bunch of mistakes here. That's why I like doing these streams, so I can learn from other people too. So please let me know if I've made any mistakes, or you guys have a better explanation for something. I would love to hear it. Seriously. So the final thing is monoclonal antibodies, which I always kind of give a very boring example of how they're made, but a lot of people already know that. What a lot of people don't know about monoclonal antibodies is how they're used. They could be used to kill things like tumours. Because the way tumours work is you could get your monoclonal antibodies, okay? Imagine they're like these things. And because you can create millions, billions, a shit ton of them, you're going to be able to have basically an endless supply of them. So they get a bunch of them, and they will attach things like a radioactive isotope, I believe, or a chemical or a poison, something like that. They will then send the antibodies right into the tumour, and it will bind with it. And when it binds, obviously the radioactive thing, it's going to kill it, or it's going to cause damage to it. That's why people assumed that monoclonal antibodies would be the cure to cancer. Of course, the issue is, they are tumours themselves. They could grow en masse, they could not work, it could make things worse, they could end up damaging your cells, who knows really, there's a lot of unforeseen consequences. They cause like hair loss and stuff, but anyways, that's one way that they work. You attach things to them and then you send them. a way that they work in things like pregnancy tests is that there is a hormone in people who is pregnant, like their urine, and the way that works is you would have a bunch of monoclonal antibodies with dye, like coloured dye. Then when this hormone is detected, the monoclonal antibodies will respond because they're lymphocytes. They're designed to activate when they get a response. They will respond and you'll get a colour change. Same way that COVID tests worked. So that's why I like biology, because it makes things that like maybe people didn't think about like a lot more obvious. It doesn't look like anybody is saying anything in chat. Like Kieran hasn't said anything in a long time. So what else should we look through? Let's go. Let me go from page 1 now. In fact, let me just look at a quick overview. We got cell biology. Oh, mitosis. Oh, I forgot about mitosis. Let's do mitosis. Okay. I prepare meals at the college. That's how I remember it. So you have, for like a pie chart. Basically, this is interphase, this is mitosis, and this is cytokinesis. You have your interphase, which is regular cell activity, except the chromosomes will replicate to be ready for mitosis. During mitosis, you have prophase, metaphase, anaphase, and telophase. Prophase is where they basically leave the nuclei, the chromosomes. The nuclear membrane is broken down, disintegrates. It's broken down. I forget the terminology. Nuclear membrane is broken down. Metaphase, they line up a bit like Facebook's like workers protesting. I don't know. I try to think of funny word associations here. Anaphase is where so the spindle fibers will attach. Anaphase is where they're pulled apart to each end. I've missed a step here. Maybe. Telophase is when new membranes are attached, new membranes surround the chromosomes, and the final step is where the cell is pinched to create two identical daughter cells. There we go. Now, meiosis, I don't know how you pronounce it, is basically the process of mitosis, except it happens again. Except this time, each one will only have half a pair of chromosomes, because obviously they're sex cells. Okay, let's keep going. Culturing microorganisms. Okay, we could do that. It's a practical after all. Let's see what I remember, and then we'll go look if needs be. This is going to be a fire here. I've drawn that abysmally. Hang on. So that's your Bunsen burner. What you would do is you would get your wire. Not sure if there's a correct term for it. Your inoculating wire. Inoculating loop. Hang on, it's something like that. Let me see if I remember. It doesn't say in my revision guide. Okay, but you get your inoculating loop. You would get a petri dish with agar jelly, which basically is the nutrients for bacteria to grow. You would swab it, like so, on the plate. Oh, I guess I should specify, you should also dip it first in your microbe solution, but then you would obviously put it around. You would then place it in like an incubator, which should be 25 degrees Celsius for like schools, because you don't want to make like a fucking COVID-50 or some shit, right? So you also shouldn't seal it, because then it encourages the bacteria to do anaerobic respiration, and those are usually the most like dangerous ones, you know, the big bastards of the bacteria world. Diffusion, osmosis, they're really fine. Well, watch me get a question tomorrow about it and get it wrong. We got stem cells. Stem cells are undifferentiated, but they can later specialize in order to become different cells. They are found in embryos, basically, because embryos are just clumps of cells. People get really pissy about it because they think that, like, they're humans. I don't really care. I'm not an embryo myself. I couldn't tell you what's what in that regard. But in other words, they're quite ethically bad in that sense. But embryo stem cells can become any type of cell. Meanwhile, bone marrow cells can only become a very select few, like blood cells, which means bone marrow are unable to treat all illnesses or diseases. Embryonic could, potentially. There are issues, however. Meanwhile, if you take someone's bone marrow and put it back into them, if you take their stem cells, put them back in, there'll be no rejection. Yeah? No rejection, because it's their own DNA. Meanwhile, if you take some Donnie's embryo and take the stem cells, you could get rejection. That's why people assume that stem cells are pretty good, but they're not as good as we think, type of thing. They're obviously useful for life, but I mean, like, curing illnesses. People thought they could cure blindness and shit, but, like, maybe they do? Who knows? Anyways, I'm gonna go take a 2-minute break, just gonna go do something, and I'll be right back. Gonna go grab a drink. Huh, okay. Anyways, like I was saying, I think we're almost done here, genuinely. If you guys want any other stuff, let me know, but let's have a look. Cell organization, enzymes, digestion, the lungs, cancer, respiration, metabolism, and exercise. Those are the ones we'll do. So what was it? It was the lungs. Let's go look at the lungs, because I'll be honest, maybe I've missed something in the lungs. Okay, the lungs are the top part of your body. The thorax is the top part of the body. The lungs, pink sponges, protected by the rib cage, surrounded by pleural membrane. The air that you breathe in goes through the trachea, splits into two bronchus, going into lungs. They split off into bronchioles, which end with the air sacs, the alveoli. The alveoli carry out gas exchange. They contain millions of air sacs. Blood capillaries allow for gas exchange. We know this. When blood reaches the oxygen, it is released from red blood cells and diffuses into the body. So when the blood reaches, the body, cells is released. Yeah, that's all right. This is okay. The blood passing through the alveoli is just returned from the lungs. It has lots of CO2, very little oxygen, so oxygen diffuses out of the alveoli, out of the blood. So carbon dioxide goes out of the blood, oxygen goes in. We know this. Is there anything else here on the lungs? Circulatory system. The right ventricle pumps deoxygenated blood to the lungs to take in oxygen. The blood then returns to the heart. Yeah, that's a double circulatory system. Right, let's go do topic 3. Let's see if there's anything here that we need to do. This is infection and response. Prokaryotes and eukaryotes. Pro or without nuclei. Eu or with. Developing drugs. Drug tests, very quickly before we remember. Drug tests. So the way it works is they're first tested on, I believe, they're first tested in computer models. The drug is tested on human cells, animal cells. They're then tested on animals. They're then tested on healthy volunteers, infected volunteers, and then finally infected. So, drugs are tested on human cells and tissues. So for example, if you're testing for like blood pressure, but you can't do that with a couple of cells, you've got to use an animal. The next step is to test it on live animals to test the efficacy. So whether the drug produces the effect you're looking for. So efficacy is how efficient it is. Will it actually work? Toxicity is how harmful it is. If you take a drug and you start, your eyes go red and you start dying, maybe it's not too good, is it? Dosage, so how much? You don't want to fucking overdose if you take like 50 shots of it. Some people think animals are so different from humans that testing is pointless. I mean, yeah, true. They have different, obviously, DNA and shit. Different, structure. If a drug passes the test on animals, it's tested on healthy human volunteers in a clinical trial. So firstly, they need to make sure it doesn't have any harmful side effects by using a little dose. Second time, it's tested on those with an illness. The optimal dose is found. And then obviously the one that's the best is the one with least side effects and most effective. Patients are put randomly into 2 groups. One is given the new drug, the other is given a placebo. This is so the doctor can see the actual difference the drug makes. It allows for the placebo effect to happen. Clinical trials are blind. The patient in the study doesn't know whether they're getting the drug or a placebo. Often they're double blind to prevent a bias. The results of the drug testing aren't published until they've been through peer review. Okay, that's all right. Light intensity is 1 over the distance squared. Okay. You can artificially create ideal conditions for photosynthesis with farming. Respiration is the process of transferring energy from a breakdown of glucose. It goes on in every cell in your body continuously. We know that. So glucose is obviously broken down. Well, glucose is used in the respiration reaction in order to create ATP energy, but it is also used to produce CO2 and water vapor, right? Respiration transfers energy for all kinds of things. So here's some examples. I guess write these down. So 3 examples of how organisms use the energy. You might get an exam question where it's like state 2 uses of the energy. Build up larger molecules from smaller ones, such as protein synthesis. In animals, it's used to allow the muscles to contract so they can move. In mammals and birds, the energy is used to keep their body temperature steady. So, I guess, regulation of temperature. Metabolism is all of the chemical reactions in an organism. Here's a 2 mark. Give 2 examples of how animals use energy. What is metabolism? We've just literally just said that. Aerobic is respiration using oxygen. It's the most effective way. Most of the reactions happen inside the mitochondria. Remember that mitochondria is for energy, powerhouse of a cell. It releases energy. Here, the word and the symbols. Okay, so it's C6H12, C6H12O6, which is glucose, plus oxygen, gives you carbon dioxide and water. We know that. Anaerobic respiration is without the oxygen, so that's obviously going to be glucose and just the lactic acid. And in plants and yeast, that's how we get bread and shit like that, because it produces ethanol and carbon dioxide. I'm going to mute myself quickly so I can be... Okay, we're back. Sorry about that. See, I already think I know this. So what are the reactants of aerobic respiration? That's obviously just going to be glucose and oxygen. What is the process of anaerobic respiration in yeast called fermentation? I remember that from like playing Minecraft like 20 years ago. OMG. Exercise, muscles need energy from respiration to contract. Oh, and obviously anaerobic respiration causes an oxygen debt because you're going to have a bunch of glucose, lactic acid. I'm bloody delusional now. Lactic acid will be in your blood. It will cause your muscles to, I guess, what's like the terminology here? What's the terminology? It doesn't really say. I think it just neutralizes the lactic acid because your oxygen is able to... Oh, it allows your liver to obviously get rid of it. Destroy it. Destroy it? What's a better terminology for that? The blood enters your muscles, transports the lactic acid to the liver. Oh, it's converted. Okay. That's fair enough then. But I think that's everything. That's literally everything. We have gone through the whole of paper 1 in like an hour and a half. Oh, someone said something. Hey, hello. I think that's it, genuinely. Again, I only looked at the book there just so I could recap myself on 1 or 2 things I wasn't sure of. But I think I'm calm. I honestly think that that's where I'm ending today's stream. I feel very confident about paper 1. I'm not going to go in and act like I'm the bee's knees. But you know what I mean? I genuinely think that it's a solid paper. There's some fun stuff, some easy stuff. I think, honestly, the things I would not look forward to is respiration, just because I feel like I've gone through that the least. Maybe the liver. Kidneys. Shit, are we meant to know about the kidneys? No, that's paper 2. Thank God, because I was going to cry. I mean, I guess you do need to know, obviously, like, waste products and stuff. Like, waste is carried through... Oh! Blood. Quickly. Blood. Blood contains 4 main components. You have your red blood cells. Red blood cells are a biconcave shape. They have hemoglobin, globulin. It's a weird word to write. Hemoglobin. You have your white blood cells, your things like your phagocytes. Oh, Jesus Christ. Your white blood cells, your lymphocytes, phagocytes. You also have plasma. And all I really know about plasma is it's used to carry things like CO2, water, and glucose. And then finally, you have one that everyone forgets, platelets, which basically clot together So if you have like an open cut, they clot your blood back together to stop it from flowing. Which is useful because imagine like, so for example, people with certain types of cancer, like maybe blood cancer or something, I don't know if that's a thing, or like cancer that like affects your blood, they may have it where if they get cut, they will just bleed non-stop, and that's because it reduces the amount of platelets in them. Anyways, let me quickly go check. What was I going to do? Oh, BBC Bite Size. I just want to check if there's any practicals that we should know before going into this exam that I haven't not done. AQR. Okay. Are there any... Let's just look at the exam questions. Let's see how many of these we can answer. Number 1. What is the part of the animal cell labelled by A? And it is literally the nucleus. Oh, these are foundation, right? Let's crank it up a bit here. Can we get higher questions? Okay. Describe the similarities and differences between the process of diffusion and osmosis. Alright. So both of them are passive, so they don't require energy. Diffusion is for, like... Actually, hang on. That's just opened my eye a tad. Let me go quickly check the actual definitions of diffusion and osmosis. Just want to make sure I don't miss out anything. Diffusion is gradual movement of particles, spreading out of particles from an area of high to low. It happens in solutions and gases. And osmosis is just like water, right? It should just be water. Movement of water across a partially permeable membrane. Yep. Okay. So both of them go from a high concentration to a low. Osmosis is water. Diffusion is solution and gas. Osmosis is a partially permeable membrane. Both of them are passive, so no energy. That's your 4 differences. 4 similarities, differences. Compare the advantages and disadvantages of light and electron microscopes. Oh, I like microscopes. As I say in every stream, I have one myself, which I never use because I don't have any solution stain for it. Anyways, advantages. Electron microscopes have a higher resolution and higher magnification. Electron microscopes provide a 3D image of whatever you're looking at. Electron microscopes are a lot more expensive. Light microscopes have a lesser magnification, lesser focus, but they are cheaper, and I mean that's kind of it really. Yeah, higher magnification, higher resolution. Examine the structure. Light microscopes can be used to examine living cells. Oh, interesting. Light microscopes can be used to examine living cells. Electrons can't. But electrons can scan the surface structure. So obviously because they're looking closer, you won't just be able to see a single thing. Right, let's keep this going. This is fun. A student is calibrating an eyepiece. The student finds 45 divisions on the eyepiece graticule correspond with 100, I'm assuming that's micrometers. Calculate what one division of the eyepiece of this magnification represents. Oh my god, it's raining like mad. What the hell? Bro, I just look outside and it's raining, like... Oh yeah. Get me some of that hydrogen. That's relatively easy. Honestly, I think I'm done. Damn. Let's have a look. Effective pH on amylase. That was on one of the past papers, so I feel like that's not coming up. But that's effective pH on amylase. So that would be the higher the pH, the more denatured. Microscopes, yep. Culturing microorganisms, yep, we've just done that. Food tests, yep. Photosynthesis, that would be the pondweed. Only one I don't know, effects of osmosis on plant tissue. I've never seen this one in my life. Free science lessons, man. Please teach me. Describe how to investigate the effects of osmosis. Wait, is this the potato plant one? When a plant cell is placed in water, water will move into the cell by osmosis. If we place it in a concentrated solution, it moves out. Okay, so you peel the potato, produce 3 cylinders of the potato with the same diameter. Measure the length, make sure each one's the same. Place each cylinder into a test tube. Different sugar solutions, one distilled, I guess for a control. You would leave them overnight. Remove the potato cylinders, roll them on a paper towel to remove surface moisture. Measure the lengths in masses. Calculate a percent change, get your change over original times 100. So in other words, the potato cylinder gains mass. In concentrated, the cylinder loses mass. Alright, that was easy. That was fine. I did not mind that at all. I thought that would have been horrible, but no, that's calm. So there you go. We're done. I will sit around here for 5-10 more minutes, just in case anybody wants to ask me anything or wants me to go through anything. But I'm calm. I think this will be a you know, it'll be difficult. Will be 1 or 2 scummy questions, but you know what? It's calm, innit? Like it compared to like paper 2. Let me go just message Kira on Discord here. So, there we go. I just asked Kieran, but I don't know if he'll reply. Let's go play some music. Where's Ezra Collective when I need it? But yeah, I'm done. I'm calm, honestly. I've never felt more ready for an exam. And I know tomorrow I'm going to end up being like, holy shit, that was horrible. But, oh man, Okay, let me just kind of wrap my brain here and think if there's anything that comes up that's bad. I can't think of anything. I genuinely can't think of anything. So, I'm calm. Hope you guys are calm. Before I go, I just want to offer some words of, like, thanks. Seriously, guys. Honestly, I know that I do these streams and not a soul apart from maybe Kieran from time to time will watch them. But thank you even if you click on just for a bit and watch. Because I do these streams for myself so I can... Because I find the best form of learning is if I'm able to explain something well enough for other people to understand. That's why I like doing these streams, you know what I mean? If I can learn, if I can understand something and I can explain it, then, you know, it is what it is. That's how I like learning stuff. The problem is these streams don't really get many views, despite the fact we are now in our GCSEs. Not many people watch them, and I think there's one or two reasons why. One, obviously, people have their own form of revision. I reckon right now there'll be 10, 20 people doing other revision streams on biology paper 1. I know that for a fact. There'll be people doing flashcards. There'll be people at school still. I don't know, probably not. But there'll be people, you know, revising in their own way. But I feel like a lot of the people that I know who I would expect to revise don't. Like, seriously, I could not tell you if Luke Kelly revises. I could not tell you if Sammy does. I know Ashton does. I know Kieran probably does. But honestly, a lot of the people that I know don't really do much revision. Or at least I don't know about it. So let me know now, guys. Do you yourself revise? Like, how much do you do? Because I know Luke Kelly almost jokes that the teachers say do 20 minutes a day of revision and I do like 2 hours. But I would argue I get more out of doing 2 hours of revision than like 2 hours of revision a week or every couple of days than 20 minutes a day. Because seriously, I was off a couple of weeks ago. I was skiving, right? And I kid you guys not, I learnt more in that time off for biology and chemistry than I did when in. I was able to teach myself the structure of a plant, enzymes, food tests, and the digestive system, and cancer. I was able to teach myself all of that stuff in literally an hour. Meanwhile, in school, I would have sat there for an hour and not known a thing. So when people kind of joke and make fun and say, oh, well, Joseph, you're only meant to do 20 minutes a day, I think, no, like, let me do as much as I want. I know when to stop myself. I know when enough is enough. And I enjoy doing revision. I enjoy doing this stuff. And I also enjoy learning stuff. You know what I mean? And literally, I said, in a span of an hour, I was able to learn myself 5-6 biology topics. Which is why I'm good at them now on stream. I know a lot of people might not like these streams, or people might criticize them by saying, all you're doing is regurgitating information that you know. You're not learning anything. But the truth is, I already know a lot of this stuff myself. If there was new content to learn and I didn't know it, I would learn it on stream. I would learn it off stream and then tell you guys on stream. But yeah, I mean, that's it, guys. Thanks for watching. And Seth.